we've got X-shaped object that we call sometimes a chromosome is really two. Are there any questions, concerns? Do we need additional clarifications on this point? Issues with interpretation of this fact? We'll go into this in much more detail as we start talking about eukaryotic chromosome structure in the upcoming classes. I wanted to start this conversation now. Yes, please.
So one chromatid is one double helix?
Yes. Okay, so for those of you that didn't hear, she asked, is one chromatid one double helix? So because you asked, we'll go into this now. I'll repeat it later. Each of these double helices, I'm going to abbreviate. There are two of them, two double helices. Somebody throw something at me when I do this. Please, honestly. Especially you in the front row, yeah. So every X-shaped object is really two double helices side by side. And each of those double helices is called, at certain points during the cell cycle, which is why we cover cell cycle again in this class, is called a chromatid. At different points in the cell cycle, we also call those chromosomes. So I apologize on behalf of all of the geneticists that came up with this terminology to just spoil the mood for you. But a chromatid and a chromosome are the same thing. They're both a double helix of DNA. It's just that sometimes the double helix is called a chromatid, some parts of the cell cycle. At other times during the cell cycle, we call the exact same molecule a chromosome. We will go through the distinctions in detail in about a week, I think, starting next class.
What about when it's called a sister chromatid? It's just the other one?
Right, so these are two sister chromatids. So that's obviously a term you've heard before. So the two of them together are referred to as sister chromatids. Why are they called sisters and not cousins or anything else? What do we know about those two molecules of DNA? Or what do you suspect about those two molecules of DNA? They're the same. Yes. In what sense?
Is it because they're replicated from the same parent?
Right. They're both copies of the same chromosome that were made from the same original double helix of DNA. We get into that when we talk about replication. But they're sister chromatids because they're identical. There are two products of DNA replication. And again, all to be gone through in more detail later. But this is just to understand when you look at a picture of a chromosome, what you're actually looking at. It's tremendously important. Other questions, concerns, clarifications? When I say chromosome, what do I mean? We didn't even start with the definition of a chromosome. Okay, so it's one DNA molecule, a double helix, and often together with proteins that help it package. Okay. So that's the definition of a chromosome. It's one double helix of DNA. And then we can discuss later whether or not we want to include whether or not the proteins that are also associated with that DNA molecule are part of the chromosome. It's what we see when we look at a chromosome in a microscope. When we look at a nucleus in a microscope. We observe an object. The word chromosome, I know this is boring for those of you that don't care about linguistics, but damn it, I make you care about linguistics. What does chromosome mean? You look back at its Greek roots. What does chromo mean? Light, color. And soma?
Body.
The body. Right, so colored body. So the chromosome was first called that when microscopists first started looking at nuclei in microscopes, and they saw things that took up certain types of chemical stains. They called them colored bodies, chromosomes. And what they were looking at was this combination of DNA and proteins that make up our chromosomes. So the goals for today are to talk very, very, very briefly about the structure of prokaryotic DNA, chromosomes, and then to move on to a little bit of terminology. We just defined chromosomes, so we're already ahead of the game. Talk about the concept of the genome briefly. Then we get into more applied genetics. Necessity versus sufficiency, so I've got a quick question and answer game for you. And then at the end of class, the last third or so, we're going to talk about a specific technique that scientists use to study chromosome structure. That's more applied genetics. So that's where we're headed today. What do you need to know about prokaryotic chromosome structure? What do you know about bacterial chromosomes? Somebody tell me one fact about a bacterial chromosome. Then we can move on. Ma'am. It's circular. Well, you didn't tell me, you asked me. Tell me. It's circular. Thank you. It's circular. So, bacterial chromosomes have no end. They're circular. So do the plasmids they carry. Notice that there's a distinction here between plasmids, which are essentially small chromosomes. They're small double-stranded pieces of DNA that lots of bacteria carry in high copy number. That is, they can have more than one version of that plasmid in their cell at a time. And then there's the much larger, what's called here, chromosomal DNA, which is a larger piece of DNA that contains all of the essential genes for bacterial survival, and it's also circular. If you want to learn more about bacterial DNA, we'll talk about it a little bit more later in the class. We also have a whole class on bacterial genetics, microbial genetics, here in the department. So if this really fascinates you by the end of the term, you love genetics, consider taking that course as well. So look, that was the whole topic for today, bacterial chromosome structure. It's circular. Awesome. So now I get to do other things, now that we've dispatched with the necessity for today. How do you know who's a man and who's a woman? In any species. Let's talk about humans. How do we define who's a male and who's a female? It's tricky. Sex chromosomes. Okay, so we can do genetics. So we could look at somebody's chromosomes and ask, well, what do you want to know about their sex chromosomes? Oh, so we've got X chromosomes, we've got Y chromosomes in humans. Who's got the Y chromosomes?
That was a mixed response.
So males
Thank you, teach.
get a Y chromosome. Okay, is this true of all species? Do all species use chromosomes genetics to figure out who's male and who's female? Nope. Okay. So we'll learn a bit about sex determination as we go along in the class. So we happen to be a species where males have a Y chromosome. But, you know, we've only been able to look at chromosomes under the microscope and actually detect the presence or absence of a Y chromosome for a really short period of human history. So aside from that, imagine you're the original biologist who is studying sex determination. You wanted to distinguish male versus female. What evidence do you use? Reproductive organs makes sense. External anatomy, breast, no breast, penis, vagina. Turns out that's not the way that most scientists define the sexes because, well, those tissues only occur in a small fraction of the total number of animals and plants on the planet. So you could cheat, read the slide. What's a really fundamental phenotype or trait that we could use to define males and females? It's the type of their gamete that they produce. So you produce a small motile gamete, one that moves, like males produce flagellated sperm. That's how we define who's a male. It's one of many definitions, but it's the most basic definition in biology. And the individuals that make the large immotile gametes that the other gametes try to migrate toward, we define as females. This is not to, especially in recent years, it's become very clear that there is no real thing as a binary sex. We don't just have pure, in any species, but especially humans, we don't have just males, we don't have just females, there's a continuum. But this is still the primary way to distinguish. If you want to make a binary distinction between sexes, you go to the gametes and ask, are they big or are they small and motile? So the most fundamental decision that an organism really makes as it goes from a single-celled organism to a multicellular organism, doesn't matter if we're talking plants, animals, whatever, is whether or not you decide to become a female or a male. And that's where, in humans, sex chromosomes come in. If you have a Y chromosome, something about that Y chromosome directs the body that you're in to become male. To have male behaviors, to develop male anatomy, to develop male hormones that circulate through the body and cause other changes that are male-like. And the first organism that sex determination and the genetic basis was figured out in was the fruit fly, Drosophila melanogaster. About a century ago, people started getting the first evidence that there was something about a gene that could cause an organism to be either male or female. Fruit flies. And this was all based on a technological advance, which was improvements in microscopy that let people actually see the chromosomes for the first time. So here in the upper right, we have a picture of what's called a polytene set of chromosomes from Drosophila. It's from their salivary glands. Early in development, this one cell type makes lots and lots and lots and lots and lots and lots of copies of the same chromosome. So they get really huge. And you can actually see them using light microscopy. And so some students that I taught in Biology 104 last year produced that micrograph of Drosophila chromosomes. So about 100 years ago, people started looking at these. When they compared the chromosomes from males and females, they could tell that there was a difference. Every time they looked at a female, which is shown down here in a cartoon form. So these are XX. Females, they had a pair of chromosomes that looked identical in shape and size. But every time they looked at a male's chromosomes, they didn't see that identity. Instead, they saw that they could match up pairs of chromosomes that looked similar, and then when they were done with that, the first pair and the second pair, the last two leftover chromosomes didn't look like they were the same shape and size. So they called that chromosome the X factor. We're not talking about singing competitions or whatever the hell the X factor is about these days. They just called it the X factor. They didn't know what it was. They didn't know it was DNA. It's 100 years ago. We didn't know the structure of DNA. We didn't know anything about DNA, really. They just knew it was a chromosome, a colored body, and the structure, the shape, the size differed between males and females perfectly. Every time they looked at a female, it looked like the shapes and sizes of chromosomes diagrammed here on the left. Every time they looked at a male, so perfect correlation between chromosome shape and the sex of the organism they took the chromosomes from. So why do I emphasize the word correlation? What else? What other C word could I use besides correlation? Pardon? Causation. Right? There's a critical difference in science between looking for a correlation and a causation. So even if you don't know anything about the process of science, what's the implied difference between something's correlated and something's causative? There's a fundamental difference. What do the words mean? How do they differ?
Correlate.
Go ahead. No, sir.
I was going to say what he said. There's a relationship for correlation. So if something's correlated, there's a relationship between the two.
So there's a relationship in a correlation. So you have a correlation between the sex of the organism. You observe this Drosophila melanogaster has sex chromosomes. So he's a male, I know it. And he also has this weird shaped chromosome. Perfect correlation. Every time you see one, you see the other. Does that mean that when you have sex chromosomes, that causes you to have a Y chromosome? Sex chromosomes being a patch of bristles on the front leg of Drosophila that they use in courtship. Females don't have them, it's just males. So is that causation? Is there a relationship there? You have this body part, therefore you have a Y chromosome? Or is the relationship the opposite? You have a Y chromosome, therefore you developed this special patch of bristles. So causation implies there's a relationship. If one thing is true, then the next thing happens. Correlation just means there's a pattern. It doesn't necessarily mean that. Here's a great example. I wish I had brought my slide, correlation versus causation. I didn't mean to get into this so deeply, but you let me wander off topic. Shame on you. I'll bring it next time if I remember. There's a perfect correlation between the sale of, or the consumption of cheese per person in the United States with the number of people that get strangled to death by their bed sheets overnight. So if you plot by year on the x-axis, per capita consumption of cheese, it goes up and it goes down. So how much cheese every person in the US eats per year, it fluctuates. Looks exactly like the same graph as the number of people that die each year by being strangled in their bed sheets. That's correlation. There's absolutely nothing to do with, oh, you ate a lot of cheese, so you died overnight by getting trapped in your bed. That's correlation. Just because a pattern exists does not mean that the two things that you're looking at are related to each other.
Correlation does not mean causation.
That's right.
So you can usually say that, but you can't say causation doesn't equal relation.
So here's an even more critical point. Geneticists. Budding experimentalists. Let's play a game. This sweater has amazing powers, actually. It's really fabulous. It makes me super strong. I got this from my daughter's cartoon. This example. No, seriously. This sweater makes me really strong. Physically. Now I'd like you to prove that I'm wrong. Who wants to challenge me? Mentally prove that I'm wrong. What sort of things would you want to know? What sort of tests would you do to test whether my sweater gives me fabulous strength? Gives whoever wears it fabulous strength. Pardon? Make me lift weights. Okay, so that would prove that while I'm wearing a sweater, I'm strong.
Yeah, because it's a difference between wearing it and not wearing it.
Okay, so you'd ask me to take off the sweater and see if there's a difference. Right? That's the one way to tell whether or not this sweater gives me superhuman strength. Now, is that just me or is that anybody? So I said, whoever wears this sweater gets to be super strong. How do you test that? So make somebody who doesn't already have the sweater put it on and see if that affects them or not. So these are two different experimental approaches, taking something away and seeing if it has an effect, and then adding that same thing to somebody who doesn't already have it and noting whether there's an effect, that are two critical parts of experimentation in most fields of biology, particularly genetics. And those two approaches define the terms necessity and sufficiency. So if you want to prove that something is necessary for a causative relationship, I'm wearing this sweater, therefore I'm strong, what do you do in that scenario? Which of those two experiments? You take it away or you give it to somebody else? To prove that my sweater is necessary for me to have super strength.
You put it on you?
You take it away from me. removed from the person, I'll say, that has it. Okay, so how do we know if my sweater is sufficient? That means it's all that's necessary. Sufficient, it's the only thing you need for you to become super strong.
Give it to someone else.
Then the person who doesn't already have it needs to receive it and test that. So sufficiency is what you find out about a gene by giving it to somebody who doesn't already have it and seeing what the effect is. So now I'm making the story a little bit more concrete. I'm not talking about sweaters, I'm talking about genes. Give a gene to an individual who was lacking it. So two important types of experiments that practically every geneticist does. If you want to know about the effect of a gene and whether there's a causative relationship, not just a correlation, if you want to know that a gene causes an effect, like super strength, you test whether or not it's necessary for that effect by getting rid of it and seeing if that effect goes away. And you test if it's sufficient to give somebody that effect by giving them that gene and finding out if that gene causes the effect that you expected to see. So let's apply that to sex chromosomes. So we have Drosophila. They've either got an X and a Y or an X and an X. How do they tell who's going to be which sex? What are the genetic differences between these two columns? What genetic aspects of biology could cause one of these columns to represent males and the other females? So XY is male, XX is female. How could the fruit flies be detecting what sex they're supposed to be based on their chromosomes?
They look at the X factor.
So what about the X factor differs between the two columns?
The appearance of the chromosome.
Well, so if I handed you a Drosophila that's a female, how did that Drosophila itself know that it was supposed to develop as a female? It looks at its chromosomes, not literally, but somehow. It assesses the chromosomes it has, and it says, I've got two X chromosomes. Therefore, I should be a female.
It represents it through the sex cones.
So it all comes back to, literally, the difference between XY versus XX. What are the differences just in the text there? Presence and absence. One sex has a Y chromosome, the other sex doesn't. Okay, so is it the Y chromosome that makes males? If so, how do females know that they're supposed to be females? If you have a Y chromosome and you're a male, then what would you expect about females?
The Y chromosome.
They don't have a Y chromosome. So one possibility in red is that you've got presence or absence of the Y chromosome dictates sex. So if you've got a Y chromosome, you become male. If you don't have a Y chromosome, then sort of the default situation is you become female. That's one way to interpret this correlation. You look at a female, they've always got two X chromosomes, no Y chromosome. If you look at a male, they've always got one X and one Y. There's one other difference genetically in the constitution of chromosomes between males and females.
When there's a pair of X's?
One sex has two X's, the other sex has one. So maybe it's the number of X chromosomes that controls whether or not you're male or female. So in the second scenario, maybe one X chromosome means you're male, where two X chromosomes means you're female. It turns out, you know the answer, right? Which one of these does Drosophila use? Maybe you don't know. We'll figure it out eventually. In humans, how does this work? Is it the number of X chromosomes, or is it the presence or absence of a Y chromosome that makes males in humans? It's the presence of the Y chromosome. So if you've got a Y chromosome, you develop as a male in humans, not all species that have X and Y chromosomes. Drosophila is actually the opposite. In Drosophila, they count the number of X chromosomes to determine who's going to be female and who's going to be male. So the fact they have a Y chromosome doesn't matter at all. Y chromosome in Drosophila actually basically does nothing. Literally, nada. So 1 X chromosome, you're male, 2, you're female, in Drosophila. How would you experimentally test this? So say that's your hypothesis. The Y chromosome causes, in humans, male sex determination. You want to find that out in Drosophila too. What 2 experiments would you do? So XY flies are male, XX flies are female. Okay, talk to your neighbor for 3 minutes. Figure out how to test necessity and sufficiency. What sort of flies would you create to test those 2 concepts? That the Y chromosome is necessary for sex determination and its maleness, and it's sufficient for maleness. So go ahead and do that for 3 minutes. See if you can come up with what you would do experimentally. What sort of flies would you look for? Okay, go.
I'm gonna erase Linda.com now.
Okay. Should be good. I think everybody got the point. So what sort of an experiment would you want to do with, you just have male and female fruit flies. You have the technology and the power to take chromosomes and move them around. What sort of mutant fruit flies would you want to make? Sir? One that doesn't have a Y chromosome at all. So, that's an XO genotype. The O stands for, it's actually a 0. We call it XO, but it's X nothing. An individual that just has 1 chromosome, it's an X, no pair. So does that test necessity or sufficiency? You take the Y chromosome away from a male. That tests whether it's necessary to have a Y chromosome to be male. Okay. Now how do you know that the Y chromosome is sufficient to turn you weaklings with my sweater into fabulously strong people, or turn a female into a male? What genotype, or how do you move chromosomes around? So do you swap? So you start with an XX fly. If you took an XX fly and you swapped out one of the X chromosomes and you put in a Y, what have you done? You know it's going to be a male because that's the chromosome type of males. So what do you have to do that's new, that's different?
Would you keep the other X and still put the Y in there?
Ah, indeed. So you'd keep both X's, and then you'd also, on top of those, add a Y chromosome. So you start with a fly that you know, by correlation, you suspect should be female, and you say, here, have a Y chromosome too, see what happens. We'll talk about these types of mutants later in the term, especially in regard to humans. So humans have been really useful for understanding how sex determination works and how genes influence that, because when something is different about us, we tend to go to our doctors and self-report. Fruit flies don't do that. So human mutants, in the study of human mutants, people that don't have genotypes that are typical, have taught us a lot about how genes control sex in humans. But today we're just talking about Drosophila. So from what I told you earlier, the number of X chromosomes dictates who is what sex. People did actually find, by chance, mutants in Drosophila that had these genotypes. They found, by chance, they happened to find that super really rare Drosophila that was lacking a Y chromosome, the XO type. And they also happened to find XXY fruit flies. So what are their sexes? What's the sex of an XO fruit fly? It's going to be male because we count X chromosomes. So in fruit flies, XO fruit flies were male, and lo and behold, when you added a Y chromosome to an otherwise female genetic constitution, didn't change her sex, which is how they discovered that the Y chromosome in Drosophila really does have no effect on sex determination. Now, in this case, they weren't doing what I described and actually physically moving chromosomes from individual to individual. They were finding mutants that had these existing conditions on their own, naturally occurring. But these days, we have genetic techniques we can use that would actually allow us to do this. Yeah.
So you're saying that because in fruit flies, you need a double X for it to be female. Does that mean if this was humans, it would end up being a female instead because you need the Y chromosome to be a male? Like that's a determining factor?
Okay, so this is, so just to be clear then, yeah. This is FF, fruit flies. In red. For comparison, what does this look like in humans? So XX is female, XY is male. But in humans, it's the Y chromosome that controls sex. So if you're XO, if you have Turner syndrome, if you only have one X chromosome and nothing else, what sex are you? You're functionally female, not often fertile, sometimes sterile, because there's a reason why we're diploid and have 2 copies of every chromosome, and when you're lacking one, sometimes bad things happen. And then what about XXY, individuals with Klinefelter syndrome? They're male, they've got a Y chromosome. They're externally and functionally males, again, sometimes sterile. So that's a key difference between Drosophila and humans. Right? The same sex chromosome genotypes, XO and XXY, are the opposite phenotypes in the 2 different species because one uses X chromosome counting, the other one uses presence or absence of the Y chromosome. Oh, the wacky things nature does. But again, the important concept, and now I've gone totally off track, which is totally fine. I appreciate your interaction and your willingness to ask questions. The important concept that we've learned about so far is how to experimentally distinguish whether or not something is necessary and something is sufficient. A gene controlling a trait. We want to be able to distinguish whether or not it's just a pattern of correlation or whether there's a causative relationship between having a gene, say, and getting a disease or a trait. Any other follow-up questions, concerns? It doesn't distract me at all. We still learn the exact same content through the entire semester. We're just doing it in a different order.
So how would you get a person with both sex characteristics?
So hermaphrodite, how do you get somebody who has some of each characteristic? So those are often, it's complicated, as you might expect. That has a lot to do with development. And that's, so what we're talking about here are cases of at fertilization from birth, or it's from conception rather. An individual has a particular sex chromosome constitution in all of their cells. So from conception, that individual always had an X chromosome and nothing else, XO, or they always had 2 X chromosomes and a Y. They were Klinefelters. And by themselves, those types of individuals I would have to do some investigation to discover whether or not, if you have Klinefelter or Turner syndrome, if you would either informally or formally be classed as being hermaphroditic. You don't produce, technically the definition of hermaphrodite is you produce both gametes, sperm and egg in the same individual. And that doesn't happen in Klinefelter syndrome or Turner syndrome. So there are very rarely humans that are actually functionally hermaphrodites. They might have half of their body looks male and has like one testis, and the other half of the body has an ovary, but they're not usually functionally hermaphroditic. But all that happens during development. So what we're talking about here is the genetics at fertilization. What chromosomes do you have? Those differences in development happen after fertilization as the body develops, as cells divide. Then certain, like half of the body gets certain signals to turn into a female. The other half of the body, for example, might get signals from other cells to turn into male tissue. If that's an interesting topic to you, I also suggest taking Biology 155, Development, which is all about what happens after fertilization. So through cell division, how do those sorts of phenotypes develop? Other questions? Yeah.
Do you know what would happen if the fruit fly would have trisomy X?
So something else we get to talk about very soon. What happens when you have too many copies of a chromosome? So does anybody know what happens when humans have trisomies? 3 copies of one chromosome instead of 2. What's the most famous one? Yes, sir? Yeah, Downs. It's okay. Raise your hand, blurt it out. Scream if you want. Down syndrome. So if you have 3 copies of chromosome 21 in humans, that's what causes Down syndrome. So it's often the case in species that if you have too many copies of a chromosome, you're sterile or it's lethal. That is, you die before you develop, you die at fertilization, or you die soon after fertilization. So it's really, really, really important that we have the correct number of chromosomes. And that's a theme that we're going to see a lot in class this semester. We'll look at a lot of examples of that. So it turns out, yes, in Drosophila, there are individuals that have 3X chromosomes, but they're not very happy. Okay. Let's see what's next. Okay, we'll get a little bit into the next topic before we wrap up. We've got about 5 more minutes. So this is a picture I took of some chromosomes. This is called a metaphase chromosome spread. I'll use that term a lot. It means that chromosomes are spread apart. And they're at metaphase, right? Part of the cell cycle where we can actually see individual, although it's kind of dark, I apologize, individual pairs of chromosomes. So this is the nuclear genome, all of the chromosomes that are in one nucleus of one cell. So this is definition of a genome. All of the chromosomes, all of the DNA that's present in a cell is what we refer to as the genome. Now, this is the nuclear genome because if I kind of draw a pretend nuclear membrane, that's all of the DNA that you would find inside the nucleus of a cell. What other DNA does a cell have that's not inside the nucleus? So, yes. Mitochondria have DNA. So if we're talking about an animal, then we have to add plus MT mitochondrial DNA. If we're talking about a plant, we have to talk about chloroplast DNA as well. So any of these, we can use these interchangeably. Normally when people in social media, in the news, talk about genome sequencing, they're really talking about the nuclear genome. But technically that also includes all of the other DNA that would be found in a cell. The plastid, chloroplast, and mitochondrial genomes as well. So this is the nuclear genome, a picture of a nuclear genome. We're going to talk about FISH as a technique. It stands for fluorescence in situ hybridization next class. But before we go, so I'm going to go back to that slide next class. Finish up with this. which hopefully will be a bit of review. This is a figure of DNA at different temperatures. Hopefully everybody's comfortable with the blue to red transition bar down there on the bottom. Cool temperatures are represented on the left, hot temperatures on the right. This is what happens to your ice cream cone in the summer in Fresno. It goes from left to right very quickly. But what happens to DNA when you heat it up? What do we call that process? Denaturation. But I will also call it the thing that happens to ice cream. Melting. So you'll hear geneticists and other types of biologists talk about DNA melting. It's exactly the same thing as denaturing. We're just talking about when you heat up DNA, a double-stranded molecule, the hydrogen bonds that hold together the two strands break, and you separate into two strands. So this is denaturation or melting. And we can use this process, and we do use this process, and I use this process every day in my research lab to help us study DNA. The first thing you need to do if you want to know the sequence of a piece of DNA is melt it. So we get no information about the sequence of nucleotides, A's, T's, G's, and C's, until we break the two strands apart, and then we can use enzymes to read the nucleotides of one strand. So that's the start of DNA sequencing, and that's also the start of this technique called FISH, fluorescence in situ hybridization. And we'll pick that up next class when we come together.